Thank you for joining us on today's Coffee Break webinar. Our topic today is Don't Miss a Beat with LEAP, an advanced arrhythmia assay. The inability to predict a drug's cardiovascular liability prior to clinical trials or launch has resulted in numerous costly late-stage drug development failures and market withdrawals. Non-cardiovascular drugs that delay cardiac repolarization have been linked to a potentially fatal arrhythmia called torsade de pointe. This delay in cardiac repolarization can be observed as a prolongation of the period between the start of the Q wave and the end of the T wave, also known as the QT interval, on the ECG. Almost all drugs that delay cardiac repolarization and cause torsade de pointe have been shown to block the repolarizing hERG potassium current. Consequently, for the last decade, cardiac safety screening has focused on evaluating the impact of drugs in hERG channel assays and in vivo QT measurements. Although these tests have been proven effective at limiting the release of drugs that are proarrhythmic, it is widely considered that extensive focus on hERG has resulted in a high failure rate in drug discovery. For example, the L-type calcium channel blocker verapamil is a potent blocker of hERG current but does not prolong the QT interval or pose a risk of TDP. There is good reason to reconsider approaches to the evaluation of drug-induced arrhythmia. Both CiPA and JICSA are initiatives with the aim of updating the existing cardiac safety testing paradigm to better evaluate arrhythmia risk and remove the need for QT studies. One proposed test of the CiPA panel is a microelectrode array assay that tracks drug-induced changes to beating heart cells in a dish. This Cardiomyocyte MEA assay offers an ideal approach when evaluating proarrhythmic indicators in vitro. Today, Dr. Daniel Millard will discuss advances in automated arrhythmia detection using LEAP technology to support next-generation cardiomyocyte MEA assays. Dr. Millard received his PhD in biomedical engineering at the Georgia Institute of Technology and specialized in neural engineering. He currently leads the discovery and implementation of advanced applications at Axion BioSystems. Dr. Millard also serves a leadership role on the CiPA myocyte core team and is a co-chair of the HESI New Tox initiative aimed at improving in vitro neuroassays for evaluating seizurogenic risk.
Thank you for that nice introduction. As Melissa just described, the CiPA assay calls for the use of stem cell-derived cardiomyocytes for the preclinical assessment of proarrhythmic risk for new drug candidates. Axion's previous participation in CiPA has demonstrated that the Maestro provides extremely reliable results for commercially available cardiomyocytes, with a high degree of site-to-site reproducibility and software tools that track the biology with high fidelity. The Cardiomyocyte MEA assay relies on the measurement and analysis of the cardiac field potential signal, which arises from the propagation of the cardiac action potential across the functional syncytium, much in the same way that the clinical ECG arises from the propagation of the cardiac action potential across the heart. From the field potential signal, clear markers for depolarization and repolarization enable the quantification of beat frequency, strength of depolarization, repolarization timing, and the detection of repolarization irregularities such as EADs, with the latter two measures being crucial for an accurate CiPA assay. Building on the success of the cardiomyocyte field potential in the CiPA Phase 1 and Phase 2 studies, new technological advances developed at Axion allow for the transformation of the field potential signal into an intracellular-like action potential signal, adding new capabilities to the in vitro cardiomyocyte MEA assay. This signal, termed the local extracellular action potential, or LEAP, is produced by transiently enhancing the cell-electrode coupling to directly change the signal recorded at the electrode, and uses the same cardiomyocytes and Axion MEA plates validated in the CiPA studies. So how does LEAP work? The theory behind LEAP is similar to that of patch clamp technique, where the recorded signal amplitude is proportional to the sealing resistance between the electrode and the cell, such that a gigaohm seal allows the faithful acquisition of the transmembrane action potential. By changing the coupling between the cells and the electrode, we are able to transition from recording a field potential signal to the LEAP signal. The resulting leap signal exhibits an amplitude between 5 and 20 millivolts and is stable for 10 to 20 plus minutes after induction. Importantly, the signal shape changes to reflect the action potential morphology, including the plateau phase of the cardiac action potential. Advances in the BioCore V4 processor and the Maestro Pro and Edge enable the faithful acquisition of low-frequency content that characterizes the plateau phase of the signal. Additionally, since the BioCore V4 processor is optimized to detect small neural or cardiac signals, the low noise and high signal amplitude lead to a signal-to-noise ratio over 1,000 for a very clean recording. For reference, this is the field potential signal from the same electrode immediately prior to induction of the leap signal, displayed on the same voltage and time scales. The leap signal affords a number of advantages for cardiac safety assays. The first is to solidify the relationship between the field potential signal and the cardiac action potential by providing a direct one-to-one mapping for important cardiac endpoints. To illustrate this concept, we have a field potential signal and a leap signal from neighboring electrodes in the same wells. If we zoom in on the field potential signal, we can clearly see how the depolarization spike in the field potential aligns with the upstroke of the leap signal. Similarly, the repolarization feature in the field potential matches the end of the plateau phase in the leap signal, with irregularities in repolarization clearly coordinated across the two signals. The size and shape of the leap signal also improve the reliability and accuracy of automated analysis. Many of the key features for cardiac safety, such as prolongation of repolarization or the occurrence of EADs, are more easily detected by automated algorithms in the leap signal. Automated detection of EADs in particular is made possible due to the extremely high signal-to-noise ratio coupled with a faithful representation of the plateau phase in the cardiac action potential. Finally, the leap signal provides a new dimension to cardiac safety assays with microelectrode array technology. As an entirely new signal type on the Maestro Pro and Edge, new leap endpoints allow quantification of action potential morphology, such as triangulation or characterization and classification of complex repolarization irregularities. These advantages are easily realized through incorporation of the leap signal into the standard cardiac safety MEA assay workflow. As illustrated here, the leap signal may be induced and acquired following a CiPA-style field potential assay, with baseline and dosed recordings collected after equilibration in the controlled environment on the Maestro Pro Edge. Then, leap induction may be performed independently but simultaneously on some or all electrodes per well. The induction phase lasts approximately 10 minutes, during which the plate remains equilibrated on the Maestro. After induction is completed, the leap signal is recorded, providing confidence in field potential measurements, improvements in automated analysis, and information on action potential morphology. The advantages of LEAP are highlighted by a few case studies in pharmacology. Here we illustrate the change in the LEAP signal in response to the block of calcium and potassium currents. Each waveform was acquired from an individual well and is referenced to the vehicle control, shown here in black. As expected, increasing concentrations of nifedipine, an L-type calcium channel blocker, progressively shortened the duration of the LEAP signal. Increasing concentrations of E4031, a blocker of the hERG potassium channel, first prolonged repolarization and then generated repolarization irregularities. Finally, combined block of calcium and potassium currents with verapamil shortened the duration of the LEAP signal and eliminated the plateau phase of the action potential. In this second case study, we use the LEAP signal to reproduce results collected with action potential recordings in dog Purkinje fiber for Tolterodine and Terodiline. Both drugs were designed for the same indication, urinary incontinence, but Terodiline was removed from the market for torsade de pointe risk. In this previous study, Tolterodine was found to prolong the action potential without changing its shape. By comparison, Terodiline induced little prolongation overall, but was associated with significant triangulation of the action potential, which is believed to be an electrophysiological precursor to torsade de pointe. Using the LEAP signal, we confirm that Tolterodine prolonged the action potential in stem cell-derived cardiomyocytes, but did not affect action potential morphology. Terodiline, however, first prolonged the action potential and then induce triangulation at higher concentrations, similar to the in vitro results from the action potential study with the dog Purkinje fiber. These two case studies illustrate the power of the LEAP signal for standard CiPA-style evaluations of ion channel pharmacology and for more advanced characterizations of proarrhythmic risk with stem cell-derived cardiomyocytes. Next steps in the application of the LEAP signal are to pair it with other Maestro technologies to continue reinventing cardiac safety assessment in vitro. Here we demonstrate the electrical pacing of the LEAP signal with integrated stimulation electrodes in the CytaView 24-well plate. Just as the LEAP signal offers multiple advantages that complement the field potential signal, combining this technology with reliable pacing will further improve the comprehensive nature of cardiomyocyte MEA assays. In conclusion, the local extracellular action potential signal adds a new dimension to standard cardiomyocyte MEA assays. LEAP is produced through a simple, label-free induction phase and is characterized by an action potential shape with high amplitude and stability on the time scale of 10 to 20 plus minutes. The addition of the LEAP signal provides a clear translation of field potential signals, improvements in automated EAD detection, and new endpoints for quantifying action potential morphology. The LEAP assay will become available for Maestro Pro and Edge users in spring 2018. I invite you to visit the Axion BioSystems website at axionbiosystems.com to learn more about the Maestro Pro and Edge and the new applications enabled by these next-generation MEA platforms. Thank you.
And that is the conclusion for today's Coffee Break webinar. If you have any questions you would like to ask regarding the research presented, or if you are interested in presenting your own research with microelectrode array technology, please forward them to coffeebreak@axionbio.com. For questions submitted for Dr. Millard, he will be in touch with you shortly. Thank you for joining in on today's Coffee Break webinar, and we look forward to seeing you again.